Good afternoon. Thank you for this kind introduction. I try to stay in the timetable. So I will be talking again about design, but a completely different type of design, like how you can engineer cells to produce sort of valuable compounds, and with which you can also address some of our, well, societal challenges. So we can look what type of challenges are to be solved. So we can talk about a cleaner environment, we can talk about energy, we can talk about health, and in all these areas actually biotechnology, and in particular yeast biotechnology, can contribute to solving this issue, or at least to some extent. So in my group, actually, we work on like replacing chemical processes with enzymatic processes to produce chemical products. We are addressing actually energy questions, producing biofuels, and we have also projects addressing health questions, not producing new drugs, but actually how we can produce the drugs cheaper, make them more affordable. And for this lecture, actually I picked the last topic, like the health-related topic, on which I will elaborate a bit more. But clearly here, biotechnology to solve this societal challenge plays really a key role, and yeast, sort of as an organism, is very vital in these processes. Okay, I think everyone knows about yeast, and using yeast as a production organism already started many thousand years ago. For example, also the ancient Egyptians were using it to brew beer, make alcohol, produce bread. This is very well known, and I assume later on we will enjoy one of these products in the reception. But it took actually around, well, from here to here, 5,000 years actually to figure out what is behind like alcohol fermentation. And only then Pasteur realized that this is yeast, so a living organism is responsible for this production of alcohol and also for the leavening of bread dough. Then maybe for our research, more relevant dates are like yeast was used to produce insulin, which previously has been extracted from pancreas, so slaughterhouse waste. Insulin had to be extracted, purified, and injected to patients. So that was actually a really important milestone in diabetes treatment. Then another important milestone is yeast was, or is still, a very excellent experimental system. It was the first organism, eukaryotic organism, which has been sequenced. And compared to today, this sequencing project took around 5 years. Nowadays, we have technology with which you can do that in a couple of days. So there were huge advancements done in the last couple of years. Then what is also, we shouldn't forget actually, yeast is the model system. And if we look at the Nobel Prizes which have been awarded during the last 15 years in the field of medicine and physiology, 5 of them have been awarded to researchers working with yeast as an experimental system. And last but not least here, now with new technologies, we also start to create synthetic genomes. We can assemble synthetic genomes, and this has also led to so-called Yeast 2.0 project, where people started to optimize a genome architecture, redesigning genomes. So clearly, yeast is a very traditional biotechnological production system, and is still, and probably still for a couple of years, the standard eukaryotic model system. Okay. So a couple of things probably we need to know. When I talk about yeast, I mean Saccharomyces cerevisiae. This is sort of the classical yeast. This is sort of a quite fast-growing organism. It's very robust. and it's only very tiny, so it has a diameter of about 5 micrometer. It has sort of different ways to live. It can reproduce in different ways, so it has a sexual and an asexual lifestyle. I already mentioned it's a very important model system, and we learned a lot about it during the last few years. Also important, if you would like to engineer an organism, you also need tools. There's many tools available which you can use to do this type of engineering. And then last but not least, many of our products which we are using, being therapeutic agents, being chemicals, being biofuels, secondary metabolites, are and still will be in the future produced with this yeast. So now I would like to introduce a bit how we engineer biology, what is needed. I first have here a picture. This is an old vintage radio disassembled. And probably people which have a background in engineering, electrical engineering, would quite readily recognize these parts here which are laid out and can exactly tell you what their function is. And you can also put these pieces together and you will get a functional radio again. A couple of years ago, a biologist asked, actually, can a biologist fix a radio? And his conclusion was, no. Actually, as biologists, we don't have really an engineering language. We are not really used to this type of problems. We don't have a common language to really do engineering work. And the second point, which I will address in a second, is here with the radio, we have clearly defined parts. You can buy them in a shop, they will work, they always function the same way. Is this also the case with the parts cells are built of? So what are the parts of a cell, or of a yeast cell in particular? So we have the genome first. This is in a way the blueprint. In yeast, or yeast contains around 6,500 genes. And it's remarkable still, 20 years after the genome has been sequenced, still 20% of these genes have unknown function or dubious functions. Okay, so this is in a way the blueprint. Then what we all know, we have the genome. This can be transcribed, producing mRNA transcripts, so-called transcriptome, which is then translated again, forming a proteome, so all the proteins. And these proteins serve then many different functions. Can be structural parts, can be active as enzymes, for example, doing catalytic reactions, so converting a substrate in a product. And at the end, well, a cell has a certain phenotype. This results in a phenotype. Importantly, all these levels can interact with each other, can cross-regulate each other, so there are feedbacks, there are regulatory loops. And then for our work, what I marked here also, proteins can be modified. And I will come to that topic later. Then, in addition, a cell is responding to an environment. And that means actually in a cell we have a variable number of transcripts of proteins and metabolites. So unlike the radio where we know there are 2,200 parts and you can assemble the radio, with the cell we have always actually varying number of parts. And this is pointed out here. So this is a very dynamic system and which can respond to intracellular and environmental clues. So, okay. Now I will quickly talk about one type of modification which is very essential actually for our work. And this is so-called protein N-glycosylation. And this is basically the addition of a sugar tree, and I marked two of them here. So these are polysaccharides in a way. which are transferred on a protein. And these modifications are essential for life. So no one of us would be here if we wouldn't have them, if this process wouldn't happen. For example, these proteins serve in recognition, and it's actually a glycan which sits on an oocyte, and you have a sperm. So for the fertilization, the sperm recognizes a glycan on the surface of a cell, and only then fertilization can take place. So from the very beginning of our life, this is very central. They play a role in higher eukaryotes like cell-cell communication. They are actually affecting how proteins or that protein can fold properly. And they also are involved in sort of a cellular quality control system. And they also sort of can stabilize proteins. The challenge with these modifications is they're not encoded in the genome. And that was is one of the reasons people have ignored this type of modifications for quite a long time. To show you that these modifications are really essential, I have here an example. And this is about a therapeutic protein, a therapeutic antibody which can be used to treat cancer. And this is a small structure, and what I mark here is an oligosaccharide. This is attached to this protein and is very essential for the function, actually. Now you can produce these antibodies decorated with two types or two different N-glycans. One might look like this, and one might look like this. And now you can test how efficiently this antibody can kill cancer cells. And this experiment showed that it is a tiny here addition to this glycan tree. So this is a small monosaccharide being present or not, which increases the activity of such a drug by almost a factor of 100. So one tiny addition of a monosaccharide has tremendous effects on the efficacy of such a therapeutic antibody. Okay, so for the next couple of minutes, I try to a bit show what my group is working on. And the example I'm talking about is production of antibody in yeast, and we can consider a cell like it is here, like it is shown here. So we have a blueprint in the nucleus. We can talk about product assembly and cellular quality control. This takes place here in the endoplasmic reticulum, one of the cellular organelles. Product processing can take place. And this is basically the reshaping of this glycan structure. And only then we have a final product which leaves the cell. What is important to note, so this product assembly line consists of around 570 different proteins which have to work together and are responsible to catalyze all these functions. And in addition, I mean, this is embedded in the cellular context, so you need energy that these processes run, you need precursors that you can produce these compounds. Okay, so one aspect we are interested in is how you can tailor this type of modifications, so adapt them or create specific structures. And in theory, like this process is conserved in eukaryotes, but there are different flavors. So for example, mammals produce different types of structures than yeasts. And what we did a couple of years ago, we started to modify these glycosylation pathways in yeast, creating also like synthetic, we called it the synthetic N-glycosylation pathways. This is shown here. So we have now a system in hand, a yeast, with which we can really tailor like these products, producing defined structures. And if you paid very good attention so far, you will see that actually this structure here is the one which I showed you on the antibody, which is really essential for the activity of antibodies. Okay, so this is, if you want to produce antibodies, this is essential, that you can modify these glycosylation patterns. Then a couple of words about like the product assembly. So we know we have the nucleus, where we have the genes, we have transcription taking place, we get RNA produced, and then this RNA is converted or is translated, and proteins are formed. And for the antibody, you have two types of protein chains. They have to come together to get the final product, and this is shown here. You have the so-called heavy and the light chain. The heavy chain is modified. They come together, and at the end, we have a structure like this, where 4 chains come together. Now, this molecule, if it's properly assembled, can leave the ER, continue in the next compartment, or what is also shown here, it can be degraded. And what is important to know now, there is actually a cellular quality control mechanism. So in this compartment, we have again a glycan-based quality control mechanism, which makes sure that only properly assembled protein is actually exported, and protein sort of which fails this quality control step will be degraded. So will be cleared from the cell and removed. Okay. So you have seen now that there is a certain space where this product assembly is taking place, and I will lead you now through a couple of experiments. So for example, we need some inspiration how we modify the cells, and you can find them also in nature. And one example we looked at is actually plasma cells, which are natural antibody-producing cells. And what you can see from that structure here, this is an electron micrograph, is that it is basically filled up with this very compartment we need for product assembly, so this endoplasmic reticulum. And we then thought, okay, can we modify the yeast also to have just this space for product assembly, so increase this compartment. And yes, this can be done. There is some complex regulation behind here, which you can inactivate, and that means that this product assembly space grows. This is shown here, and then that is also correlates with increased production. So if you increase this product assembly space, you have it here, you can increase productivity of such a cell quite tremendously, 3 to 4 times. What you also can do, and there's many proteins actually assisting in protein folding, we have now sort of like a bigger space, we can functionalize this. So you can try to add here different folding catalysts. You can optimize their expression levels. So these are proteins assisting in folding. And you can again identify constellations where you can increase productivity. So functionalizing this folding state. Then I mentioned before, like there is quality control system in a cell, and that's also active in our bodies. Actually, if you wouldn't have it, and some people might actually at some point develop a problem, like then you develop Alzheimer's disease. This is actually when the cellular quality control system starts to fail and cannot remove any more misfolded proteins. And this has also a very important impact in our system. So we can modify it, you can remove certain proteins, and this can again either positively or negatively affect productivity of a cell. So this is shown here. And again, this is actually based on these glycans. Then last but not least, I told you 80% of the genome, of the yeast genome, we know exactly what it is coding for, what the proteins do, but still 20% is unknown. So to really optimize a system, you have actually to understand everything, or you have to try to figure out how these 20 or what these 20% do and can contribute. So what we actually did a couple of, or last year, during the last years, is we created yeast, a yeast strain collection where we could delete each gene individually. And then we could probe how this affects production of a target protein here, an antibody. As we have seen this, we have a platform where we can test this clone. This is automated, sort of a robotic platform. And then we can analyze what individual mutations do to productivity. And I just mark here a couple of hits. So this is the outcome of a screen. So you have mutations which increase productivity. So everything here above the baseline. And there are proteins actually which we would expect to have this effect, this positive effect, like these two. Or you have then also, you find genes, if you delete them, which have a negative effect and reduce production. So we learn more how the yeast is working and what these proteins or what these proteins might, in what process this protein might be involved in. So, summing this up, I try to introduce you a bit like that cells are very complex systems. I hope I could convince you that this protein modification is very essential. And what I showed you with the examples that we can generate really these highly specialized cells for production of antibodies, which maybe or hopefully in the future might contribute to lower like the health costs. What I would also like to have shown you here is that designing and this engineering of cellular factories, that this really remains a challenge also in the future. Then last but not least, actually I would like to thank my research group. Some of them are sitting in the back. So without them, this project wouldn't have been possible, this work wouldn't have been conducted. So I'm really grateful for their contribution. And then also I would like to acknowledge like the collaborators and funding bodies. Thank you.